everyone. Thank you for joining today's webinar on the integrated chemical environment. My name is Amy Klippinger. I'm the director of the PETA International Science Consortium, and I'll be hosting today's webinar. Today, you'll hear an overview and see a demonstration of the integrated chemical environment, which many of you know is a publicly available web-based resource that provides curated data and multiple tools to facilitate the safety assessment of chemicals. This resource is regularly updated, and our speaker will highlight some of the updates that were released as recently as yesterday. Before I introduce our speaker, I'd like to point out that the slides from today's webinar can be found online at the link on the screen, and a recording will be posted there as well shortly after the webinar. Following the presentation, there will be time for questions. Everyone is on mute, but you can type any questions that you have in the questions section of your GoToWebinar toolbar, which you should see on the right-hand side of your screen. And we'll save questions for the end of the presentation, but do type them in as soon as you have them. So our speaker today is Dr. Shannon Bell from Integrated Laboratory Systems, working on a contract supporting the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, or more simply, NICETM. For those of you who may not be familiar with NICETM, it is an office within the National Institute of Environmental Health Sciences division of the National Toxicology Program. And NICETM provides technical and scientific support for ICVAM, which is the Interagency Coordinating Committee for the Validation of Alternative Methods. Shannon is the technical lead for the Integrated Chemical Environment, and she heads the CompTox and Data Science Group at ILS. And with that, Shannon, I'll turn it over to you, and you can go ahead and get started.
Thank you, Amy. I'm really excited to go over the update today of the integrated chemical environment. We've been out for about 2 years now, and we're continually making updates. We're always interested in hearing feedback, so please feel free to email me or contact ICE support, and we'll get in touch with you. So let me turn my pointer on. There we go, and get started. So first I'm going to acknowledge the team. So ICE has been a huge effort, and there's a couple folks well, more than a couple folks who spent a lot of their time and resources contributing here. So I'd like to recognize those, especially one of my counterparts over at SciOM, Jason Phillips. He helps lead the back-end information management, and if you are interested in going there now while I'm talking, feel free. The URL is down at the bottom. But this has been a really great effort from most of the NICETM team, has contributed in some way, shape, or form over the past couple of years. So Amy did highlight a little bit of what we do at NICETM, but the important things in terms of ICE are that NICETM really helps to evaluate alternatives to animal use for toxicity testing with the idea that we're looking to replace, reduce, or refine existing methods. A lot of the tools that we develop and the data sets that are developed are in support of ICVAM. We also conduct and publish a lot of analyses and evaluations of data that use the new testing approaches. And so, as you can imagine, we have a lot of data that could be of interest to a large group of folks who are interested in addressing the alternative test method development. So we created the Integrated Chemical Environment, or ICE. This resource is aimed at being an access point for NICETM stakeholders, and so this includes risk assessors, chemical producers, and method developers. We wanted one central spot where folks could have a very user-friendly access to the data that we have high confidence in. So a lot of the data that's used in these validation studies as well as some reference chemical lists. And so those are the kinds of resources that become very important as a method developer, whether it's a computational method or an in vitro method. You want to have data that you feel good about when developing your models and putting, evaluations of them in place. We wanted to create an easy-to-use resource, and I hope we have that, that really supports the prioritization and exploration of data. So in that vein, we have two different tools. The first one is search, and we were aiming for an ease of use and organization of the data by regulatory endpoints. And over the course of the next few years, you will see data being added and hopefully more organization to really allow folks who have limited background in regulatory testing or in toxicological testing have better access to that data. We also have tools that are really aimed at data exploration and understanding some of the ways in which tox tools are being used for safety assessments. And the goal with this is to make it easier for folks without a computational toxicology background or resource in-house to get a better feel for how these tools might be used and what resources they might bring to whatever workflow that particular toxicologist has. Why should folks use ICE? The real use of ICE is on bulk data. So everything is aimed at doing an investigation on a group of chemicals versus a single chemical at a time. We have uses for test method developers who are looking to find chemicals to serve as controls as they develop an assay, that are looking to explore whatever method they developed domain of applicability. So to understand what types of chemicals may work or may be a little bit more challenging to work with the particular in vitro or in silico assay that you're developing. For modelers, we always want data that can be used in our model development. We often, as somebody with a computational background, cleaning up data takes an awful lot of my time. So providing a data resource to the community that has data that's fairly well cleaned and standardized, we're hoping will save folks a lot of time in getting the data and getting those models out. We also include chemical parameters, and I'll talk a little bit about those later, that can be used for including more different chemicals in your existing models. With the regulatory scientists, we're really looking to help them understand potential risks of a target compound by providing some of these tools, like I said, and gather data that's out there for comparison, whether they're wanting to compare available data on some alternative methods to the standard animal method, or whether they're looking to compare the outcomes of different chemicals for the same assay. Some of the features, again, the bulk query mindset is really strong here. We're wanting to allow folks to investigate lots of chemicals across multiple different assays and data types. So as you'll see in the search, we're expecting that you're looking to get lots of different data on a lot of different chemicals at once, and we're trying to support that. We also have, for those who aren't familiar with it, fair data standards in mind. We are striving to have our data resource, meet all those standards, which means it's findable, accessible, interoperable, and reusable. Data should be cleaned, curated, and ready to use. And so the level of curation on the different data sets will vary, and we are looking at ways to communicate that better to our user base. But you will find that the data that's in ICE has gone through at least 2 levels of curation to make sure that the units are relatively standardized and that the chemical identifiers are as accurate as possible. We have some different tools to support the data exploration, and I'll be going through those tools in the live demo part. And we do continually develop and update ICE based upon stakeholder feedback. And all those listening are included in our stakeholder base. So we're really interested in hearing feedback from you on ways that we can improve its use for the community. When we talk about the data that goes into ICE, our data sources are varied. We have a lot of the validation studies that are done with ICVAN, and so these are generally pretty high confidence data, things that were done under GLP or GLP-like conditions, or were done under regulatory guideline-like conditions. We do take data from the literature base, so data that has been published that has been of use or we deem could be useful to our users. We have outputs from computational models and other decision trees, and we also do pull in data from other databases after brief curation or cleanup that we think will be useful or complementary to the data that we already have in ICE. Currently in ICE, we have data on a list of regulatory endpoints, including all the different six-pack data. The number of chemicals is approximate. Again, our data is being updated. A couple things I want to highlight is that we will be having additional offer predictions for Compara and SURAP, which are consensus models for the androgen receptor and the estrogen receptor. And you can find some details on offer predictions by going to the GitHub URL down at the bottom. We are always interested in knowing about additional data that folks have. In vivo data is very helpful, especially if it is generated using some regulatory guideline-like requirements. And you'll see that we use it in the in vivo to in vitro extrapolation tool to help give user feedback on how well their assays are doing in estimating in vivo exposure. And on that same vein, we're very much interested in toxicokinetic data. That data can be useful to a variety of our stakeholders. So we have three main tools currently in ICE, and the focus of these tools is really for data exploration and providing access to some of the computational approaches. While this is an ongoing area of development, we do have some pretty mature products out now. The IVIVE tool I'll go into in a little bit. Well, I'll go into all these in a little bit, but this one I'll spend a fair bit of time on. And that allows users to go from a response in an in vitro assay and estimate what the in vivo exposure may have been. We have a machine learning tool, which is really for data exploration and starting to understand how one can build some machine learning models using different data types, and then a chemical characterization tool, which is an area of active development. So I'm going to switch over to the live demo now. Excuse me. Again, this is a publicly available resource, so you need no password, no login to access it. The first thing you'll notice on the home page is we have a section here for news and events. When we do updates and new releases or any meetings that we may be presenting at, we'll try and make sure that is communicated here so that you can always find out about the changes that may impact your workflow. If you're interested in learning a little bit more about how to use different tools, we do have a help section. The help has user guides for the different tools, frequently asked questions, and how to contact us. We also have a data section that gives more explanation about the different data sets that are in ICE and describes in more detail about the chemical quick list, which I will be going over in a minute. So the first thing I want to turn your attention to is the search. So we're going to go over here to search. You'll see a pretty simple screen. There's a couple different options, but we're going to go over here and select assays. So like I said, the data in ICE is really organized around regulatory endpoints. So if you hit the drop-down list, you'll see that there's several different regulatory endpoints: acute oral toxicity, skin sensitization, and then at the bottom, you'll see Tox21 and PhysChem. Tox21 is a curated set of the Tox21 data, which includes ToxCast data. And this has been curated to remove any chemicals that had questionable or failing QC grades, as well as any assays, particular chemical assay endpoints, where there were flags raised that would lead us to question the reliability of that particular call. And you can get more detail about these in the under data, our data sets explanations. So I'm going to go through here and do a quick query. Endocrine, we happen to have quite a bit of data here on that endpoint. So you can see it's organized in collapsible menus. Data that's coming from Tox21 is indicated as such because we do have some different groupings there, which I'll go into more detail. But we can go and we can either select all, and you'll see them propagate over here. We can remove our selections and then select them individually. So selecting this top level group brings all the stuff down here. If I want, I can go and then add more. So let's go over to Tox21. You'll see that because I already had this selected in my prior list, it's already selected for me, so I know that's there. And I can go ahead and select a few other assays. So what we've done for the Tox21 data is that's several different assays. They have been grouped based upon the assay target. So androgen receptor pathway are assays that probe the androgen, different parts of the androgen receptor pathway. Cell cycle are all assays that measure some components of the cell cycle. And again, explanations of these can be found under our data sets information over here. The PhysChem includes both physicochemical properties as well as some ADME properties that can be useful in modeling. So, for example, the intrinsic clearance and fraction unbound measurements are useful for IV/IVE analysis, and so we've made those available here. So as you select, you can see that over here your list is propagated. Once you're done selecting, you can either run your search right now or you can further refine it. To refine your list, you can add in some chemicals by typing them into this chemical box over here. Or if you're like me and you're really bad about remembering chemical names, you can go over here and select chemicals from our chemical quick list. Chemical quick lists are described in more detail under data, chemical quick lists, but what they are is they're lists of chemicals. We have two different types. One are reference chemicals, and those are chemicals that were chemical lists that were developed for a specific endpoint in mind, and so they not only had the chemical, but they had information on the assay and the response for that chemical in that assay that can be useful in determining how well that assay performs. So, for example, the AR in vitro agonist reference list is a collection of chemicals that were probing the androgen receptor for an in vitro assay in agonist mode. And so we not only have the data supporting their inclusion in the reference list available as a download, but we have the reference available too. And so if others were looking to probe the androgen receptor on an in vitro assay, this list may or may not be suitable for your particular assay, but is generally informative on that particular relationship. So you can go ahead and select as many lists as you want, and those will get added here. You can also at the same time add in your chemicals and as many as you want. It will only accept CASRNs at this time, and if you put in a CASRN that doesn't exist, what will happen when you run your search is you will get an error that it was not found. We have a couple different features for expanding your chemical search or narrowing it. You have the ability to toggle between choosing the union or the intersection. Union is, think of it as and criteria. So I want all the chemicals that have log P data and have intrinsic clearance data and have Tox21 cytotoxicity data, versus intersection, or sorry, union is or. So it's very permissive. Intersection is the and logic. And so you can see where if I say, oh, I want stuff that has log P and intrinsic clearance, you're going to end up with very, very few assays at the end. versus if you go ahead and do union, which has or criteria, you'll be able to get a lot more. So I'm looking for stuff that has log P or intrinsic clearance or cytotoxicity. And so we default to union to make it more permissive, but if you have a really stringent criteria in mind, go ahead and change that to intersection. We've also added the ability to add chemicals that have identical QSAR structures. And so you'll see that indicated as QSAR match in the results. So QSAR structure, that stands for quantitative structure activity relationships. So think of it as a chemical structure that has been flattened and desalted. So all those additional salts have been removed, and you're just looking at the simple structure. And that's often how chemicals are processed for QSAR modeling. As you can imagine, when chemicals get tested in assays, different salt forms are often used, and those different salt forms have very different responses. By adding in chemicals that have identical QSAR matches, you're able to query all the different salt forms that may have different in vitro or in vivo measures, but would end up having very similar in silico measures if they used some kind of chemical structure prediction that used a desalted form. So I'm going to go ahead and run the search now, and it takes a minute to process the query, but in the end, you're given the details, which tells you details of your query, including all the list here of different ones. You can choose to probe these a little more or go back and edit your results. What you can see here is you have your big table with all your results, and this is a summary view. So, for example, with the Tox21, this is a summary of 34 different assays that are included in the cytotoxicity grouping. We can see how many were active, how many were inactive, how many were flag omit, and that means that when we integrated the flags, certain flags were thrown that made us feel like the data was questionable and therefore was excluded. If there were any that were untested, you will see the number that were untested. If any were QC omit, you will see that found here as well. And QC has to do with the chemical QC. So you can go ahead and filter on the different ones. Like I mentioned, there's the QSAR match option right there, and this QSAR ready ID is the desalted smalls code. And so if you see right here, these are two different two different forms of the same chemical. These both have the same QSAR ready structure, and so while they may have very different responses in the different assays, they may be informative. So in this case, this one was QC omitted due to an issue of the chemical not being the chemical that was thought to be tested, but we do have data on this chemical. So it may be that you want to include this one in your further analyses instead of going with the one that was omitted. You can download your data here that gives you the summary view, which is similar to this table view over here. You can download a detailed view, which gives you all the information as opposed to an aggregate summary of the information for each chemical and assay pair that you went ahead and queried. You can also click this button to query your mixtures. So you're sending this query over here over to our mixtures page. Our mixtures is what it sounds like. It's data on chemical mixtures. Currently, it's mostly formulations having to do with the six-pack. The way this data here is organized is you will have your formulation identifier, you have the different active ingredients, and that's where those chemicals are matched. You can see the percentage and then the information on the formulation itself. So we will be continuing to grow this out to make this a little bit more useful and give some examples, hopefully by the end of the year. We do end up getting a lot of information on mixtures, and we know that that's an active area of interest for a lot of our stakeholders is how to deal with mixture information. I'm going to switch over now to the tools, which I think will be one of the more interesting parts of ICE. We have 3 different tools. I'm going to start with chemical characterization and work my way back so I can spend a little more time on the IVIVE tool. The purpose of chemical characterization is to allow folks to look at property distributions between one or two different chemical lists. So it's really aimed at allowing folks to explore the property distribution relationships, look at what properties might be driving differences in performance of chemicals in their assays, and characterize differences between lists in efforts to either reduce redundancy or define a range of property coverage in preparation for testing. So we know that some assays just don't do well with highly volatile chemicals, and so we want to go ahead and limit those. So you do not have to select two lists, but same as before, you can either select your chemicals or enter in the different CAS numbers over here. If I select two different lists, then I will be able to see when I have my results, how those two different lists contrast. If I don't have the second list, then the orange series will just be omitted from the results. So here's a table that summarizes the data, and you can see here what list it was in. I can go ahead and filter it to just a single list or both. If I choose to download using either one of those buttons, it's downloading only the data that has been filtered down. So if I filter down to chemical list 2, that's what is being displayed in my table. So you can go ahead and see this updated. Down here is where you're getting a summary of all the different phys-chem parameters, what the distribution of, as now it's both lists, looks like. You have up here the values that are not returned, and for more details, again, you can go over and look at the GitHub page for further details on Opera. Down here is the fun part, where you get the graphs. And these graphics are fairly interactable. You can mouse over to identify what CAS RNs are represented by the different plots, and you can go and look at the different properties that are available. So currently this is restricted to Opera predictions or calculations. We can see that between these two lists, the orange series, our chemical input 1, is solidly in the bulk, vast majority of the phys-chem properties we have available in ICE. But the chemical list 2 has a lot more expansion and starts having more in the tails. If we notice that there was a difference in performance, we may look at the two different lists, and log P is one of those parameters that's often used for modeling purposes, and we can see that there is definitely a difference in the distribution between those two. Again, when you're trying to build out your particular assay, if you wanted to avoid highly volatile chemicals, this would be a way to identify which chemicals you may want to not include in that assay. So we're going to move now to machine learning. And machine learning is a pretty simple data exploration tool where you can build some machine learning algorithms, and that's where your machine learns based upon the data that you provided how to predict an outcome. And so the outcomes are limited to a couple of classification and regression ones where we feel like we have ample data in ICE to run the models, and this will be updated as we get some more data in there. I can go ahead and select an endpoint. So for example, I know I've got a fair bit of data having to do with the estrogen receptor pathway, so I'm going to go ahead and select the eutotrophic data set. Then I have to select my endpoints for modeling. And so in this case, I'm going to go here, same as before, but my endpoints are looking a little bit different. So when I go here, I'm going to go ahead and select some phys-chem properties. I maybe want to select intrinsic clearance and log P. You can see it being updated over here. Now I know that I'm trying to predict eutotrophic assay, and I might know it has something to do with estrogen receptor. So I'm going to go and pick my different endpoints. I'm going to click a couple and get some in there. Let's do some Tox21 data. So the nice thing about this is that we will do some imputation, and that means if we don't have sufficient data we will go ahead and make our best guesses, but there are limits to how well that works. And so we encourage folks to include either some Tox21 data or some of the PhysChem parameters where we do have quite a bit of data there in order to run their assays. Now, you'll be able to choose a selection of different machine learning methods. For more details on these methods, go again to the help user guides, and that will give an explanation of the different methods. And then let's go ahead and select some chemicals. So the chemicals here are the chemicals we want to generate predictions for. One of the important things to remember is that in order to build a machine learning model, you need training data. So our training data here in this case is all the data that's available in ICE that we have a eutrophic call for, so all the in vivo data. So we will add those chemicals automatically to your query in order to serve as a training set. But if you want to actually run predictions, you need to be selecting some data. Now that we've specified our assay, specified our input, specified the chemicals we want to predict and the models we want to use, we can go ahead and run our tool. So I've had this already run, but what you'll find is it gets a download folder that has a couple different items here. You have the data that went into your machine learning model. You have your user data. You have your summary report and your predictions with your test data. So I'm going to go ahead and pull up the summary report now. And what this looks like is it does a little bit of a description of the data processing to let you know how many chemicals removed, if there were any assays that had to be removed due to insufficient data. In this case, there were none. It gives you the percentage of the user-provided chemicals that were available to do the modeling. And then it goes in and gives you some summary statistics of how the models perform during the model development phase. So this does a cross-fold validation. And again, more of this information is available in the user guide. But this gives you some summary information as to how well your model was built. Some of the nice details you do get are some plots, and so you can see how the importance of the different variables in developing the models. So for the KNN model, the ER pathway model score was one of the most important variables, and the same with the SVM radial. With some of the different approaches, you may notice that one particular assay may be more informative for one different algorithm. The other plot you will get out is the model performance during the training phase. And so you can see how the ROC is, the sensitivity and the specificity of the different models that you selected. And then, of course, you'll also get a data table that has your predictions with your results. And so all this is available as a single zip file that you'll download and then have to open. So the last tool I want to talk about is the IVIVE tool. And so the goals with the IVIVE tool is that this is a computational approach that calculates an equivalent in vivo administered dose based upon the in vitro response concentration. So you're taking the concentration that gave you your ACC or your AC50 in an in vitro assay, and then you're going to go and predict what kind of exposure that would have resulted in. So same as before, you have some chemical selections and assay selections. In this case, though, your assay selection is limited to the Tox21 assays, and that's simply so that we have sufficient data to do the modeling. If you are interested in running any of these tools on your own, on the NIEHS GitHub, we actually have the tools available, and details about that is available in the user guide section. So I'm going to go ahead and start filling this out and talk through the different components here. So when we go over to assays, in this case, I am going to select chemicals first, and I want to do some predictions on some estrogen receptor or hormone type chemicals, and I'm going to choose quite a few of them. And so those have all been added. I'm going to come over here and choose my assays, so you can always find out more detail in the data sets about the assays, but I just want to get a few on here to really give us some full data. Now, when it comes to assay endpoints, you have two choices. You have your choice between using an AC50 or an ACC, and that's the response IA that came up. There's different species. We have human and rat. If you choose rat and we do not have values for rat, human values will be used instead that are corrected to account for the different sizes between the two individuals. You have two different routes of administration currently. We have in vitro, or we have an injection and oral. So we do plan on expanding this. Hopefully we will have an update this fall that allows for some additional routes of exposure. We have dosing intervals and number of days of exposure, and these are only relevant depending upon the model you choose. So there's 3 models that are available. You have a simple 1-compartment model that is based upon a single bolus dose exposure, modeling that. We have the solved 3-compartment model, and this is actually one of the models from the US EPA's HTTK package. So we've just provided a user interface, and it leverages the data that is in the ICE database for calculating a lot of the modeling parameters that are used. We also have a model that includes a little bit extra glucuronidation that's still in beta, and this one tends to be a lot longer. And like I said, it isn't in beta, so we're still working out some bugs in that, but we have it out to, as that is part of the notebook that's available on the GitHub. So for demoing purposes, I'm just going to go with the 1-compartment model. Again, this dosing interval and number of days of exposure is not relevant with that particular one, but this is nice because it is very fast. So we'll see all the results being calculated of all those different chemicals that I chose. The first thing you're going to find is you get a summary of your results, so you can download the input, you can download the results, you can download all the CASRNs that weren't returned in the query, so anything I put in that didn't have data for. This will only work on data where we have the in vitro response. So if a chemical was excluded from our instance of the Tox21 data due to issues with QC, it won't end up showing up in this particular results table. So here is going to be your summary of the data. It gives some of the measurements that were used in the modeling. You have the EAD values. Down here, you're going to have, and EAD is the estimated administered dose, so that's actually the dose you're interested in. Right here, you're going to be able to see the box and whisker plots of the EAD for all the individual chemicals running across the bottom, and the units here in milligrams per kilograms per day. Note that default here is a log access. You can toggle that back and forth, but we found it's much easier to view in log scale. So coming over here, you have the option to overlay some in vivo data. So if there's in vivo data available in the ICE database, you can go ahead and overlay it. In this case, I'm going to overlay the user trophic data. So you can see that while some assays, predictions of EAD tend to be quite good, other ones not so much. And so this whole chemical, you can see the distribution here is quite large for bisphenol B. And get more details there. You can also download the picture using any of these tools up here. The bottom chart is just showing the range of the AC50 values for the individual chemical. So if you recall, the assays that I chose were quite varied, and they may not be the most appropriate assays for looking at something that is an ER endpoint, so the uterotrophic endpoint. So we can go ahead and filter the assays. If you happen to know specifically the assays you're interested in, you can always filter by name. I tend to be lazy and use ERs or words in our search. I can also filter down on the Tox21. You can go ahead and, of course, always remove or filter down, whatever you want on this. And when you close it, the box and whisker plots and the plots below will be updated based upon that filtered criteria. So you can see it may be a little bit better for some on the overlap between the in vivo data that we have available in ICE and the predictions coming out of IVIVE. If we were to change it, though, to something different, say acute oral toxicity, we can see that the predictions are not very good at all. We get a much more conservative measurement using our IVIVE results than we would otherwise. And that makes sense given that these assays are looking at uterotrophic or estrogen receptor binding activity, and the estrogen receptor binding activity may or may not accurately reflect whether or not something's going to cause an oral toxicity. So those are the highlights of the ICE tool. I wanted to save some time for questions. I'm going to pop back up the contact information and the URL. So please feel free to go ahead and send an email if you don't have a chance to think of your questions right now. So, Amy, if we want to go switch over to the questions, if anyone has any, I'm ready.
Yeah, great. Thank you, Shannon. That was a great presentation. And there is one question so far, which is whether there's an acronym glossary to go with this database, and specifically what OPERA stands for.
We have tooltips. So let me go ahead and actually show some of the data here. So we do have tooltips in the documentation, so you can find out. OPERA stands for the Open Structure Activity Proper Relationship Application. And so Kamel has done a really nice job on the OPERA app. It includes the GUI version. So if you are interested in running these predictions on your chemicals, he has done a ton of work making this pretty user-friendly. But we don't at this time have a glossary. I can have that added and probably add that in next week.
Great, thanks, Shannon. There's a couple more questions now. If you wanted more information about the assays, where would you go on the site to find information?
Okay, so you go to the datasets page, and I'm here now. So where you can find information about where the data comes is going to be on the datasets page. So it's datasets will take you over here. And so you can get some detailed information on the endpoints, and so what the different endpoints mean. You can also get some information on where the data were obtained. So for example, with the Tox21 data, since we did some additional curation, that is noted here, the assay groupings, and so you can download some information there on how to achieve it as well. So hopefully that answers the question. And if there ends up being some further questions on a particular assay, please let us know. And if that data isn't readily available that we can point you to on our site, we will update that page to make sure that it is.
Okay. Next question is, for the TK models for EAD, are in vivo data that are available for the subject compound input to the model?
No. So for the IVIVE analysis, it just takes the different phys, chem, and ADME parameters that we have on the chemicals, and then the in vitro assays coming from Tox21 to do the predictions. The only time that the in vivo values are used is for doing the overlay in the results. So, yeah.
Another question here, but just as a reminder, a recording of this presentation and the Q&A session will be online after this presentation. Next question, what is the source of the chemical properties data?
Okay. The chemical property data that we have in ICE currently all comes from OPERA. So Opera has several different models. So since I have this open, I can run over and show you. So they've got several different models, and so you can find details on the individual models and here or on the ICE data page, where we go into the details. But currently, all of them are coming from the Opera suite of predictions.
Okay, and then I think there's a question that's in follow-up to one of the previous questions, which is, are the outputs of the TK models in the report?
Yes.
And a follow-up on that as well, one more part of it. And can custom TK models be included to adjust EAD results?
So you will get all the parameters and all the data that was used for running the models that you could then take and run on your own. You will also get the output, or rather the input that was used to the different tools, which you could take to the standalone version that we have if you wanted to modify that code. We will be bringing online the fraction unbound, two different adjustments. So one of them is accounting for the fraction unbound, the other is the Armitage model adjustment, if you're familiar with either of those. But those will be added too. But as far as a user-supplied adjustment or user-supplied modification of TK parameters, you're going to want to go over to the notebook, and I'm going to apologize because it has not been updated on the NIHS GitHub, but it is available. So this is the machine learning pipeline, and then there's our one compartment IVIBE model. We'll be updating this with just a single one that has all the models for IVIBE probably by the end of this week. And so you could take that, our notebook, and update it as you saw fit, or modify it as you saw fit for your own personal model.
All right, thanks, Shannon. One more here for now. How do you handle chemicals which can exist in more than one tautomeric form when predicting phys-chem properties?
So the way it works is chemicals, and that's one of the reasons why for the search we added the QSAR match option. When chemicals go forth for doing QSAR predictions, the vast majority of the time they are desalted and they're, I call it flattened. And I'm not a cheminformaticist, so I'm probably totally ruining and causing everyone who is to cringe. But that removes a lot of the 3D structural nature that would give like an L versus R conformation. And while we do know that that plays a role in the bioactivity, the trade-off in most cases from doing a 3D modeling versus a simplified 2D modeling is in the benefit of ignoring that. So yes, I do know it makes a difference, and I've had issues in really bad model performance because of that oversight. That is how it's handled. The 3D structure and salts do get ignored for QSAR predictions, and so you will end up with the very same QSAR prediction, at least for the Opera models that aren't composite models, for chemicals that are different salt forms. Does that make sense, hopefully?
I don't see any more questions, so hopefully that answered it.
Yeah. And if anyone does have questions, you know, feel free to send either me an email or send an email to ICE support, and we will get back to you with a response.
Okay, great. And I am just going to really quickly show my screen again, which has the web address where this recording will be posted. So if anyone's interested, you can find it there on our website, and you should also receive an email with that link in it as well. And so with that, we will end. And thank you, Shannon, so much for presenting, and to everyone for joining today's webinar. And enjoy the rest of your day. Thank you.
Thank you.